Hello friends, welcome back to my channel Medicine of 101. Today we'll talk about type 4 hypersensitivity reactions. Okay, I have already uploaded the video based on type 1, type 2 and type 3, where I have already explained you guys that type 1, type 2 and type 3 hypersensitivity, they are antibody mediated. And I did mention about this thing about the type 4 hypersensitivity there, that type 4 hypersensitivity, it will be always cell mediated. Okay, sorry. So we can write it over here. The type 4 hypersensitivity, there is a cell mediated, right? Here we get the involvement of the immune cell. So cell mediated. So how can we define the type 4 hypersensitivity? We can define the type 4 hypersensitivity in a line that is the cell mediated hypersensitivity reactions, or we can say the cell mediated tissue damaging reactions. Okay, cell mediated. Okay, let's write it over here. So you can write immune cell mediated, immune cell mediated tissue damaging reaction. Tissue damaging reaction. Okay. Now we can classify the type for hypersensitivity into two parts. In the two types, one it will be the delayed type of the hypersensitivity reactions, and other will be the T cell dependent, or you can say that what T lymphocyte mediated hypersensitivity reactions. So if you write it over here, the type of the hypersensitivity reactions. Okay, so I'm writing it over here. That's the type 4 hypersensitivity reactions. We can classify in the two ways. One, you can write it over here. That's the delayed type hypersensitivity reactions. Another we can classify in terms of T cell mediated hypersensitivity reaction. T cell mediated hypersensitivity reaction. Today we'll talk about the delayed type of the hypersensitivity reaction. In these cases, we will see the the result. For example, we are injecting the injecting the tuberculin antigens. Okay, the tuberculous antigens, and whatever the manifestations will get the changes within terms of the different kinds of the manifestations. That will take time around 48 to 72 hours in these cases. So we'll talk about in details about the delayed type hypersensitivity reactions. Then we'll go back to the cell mediated hypersensitivity reactions. Now in these cases, I am just taking the example for this thing. I am just taking the tubercular bacillus antigens. Tubercular bacillus antigens I am taking over here. Now for example, for whatever the regions, any individual who has acquired the TB antigens or tubercular bacillus antigens from the environment. So whatever thing do you know that whenever the individual who is acquiring the antigens from the environment, tubercular antigen, as we know, this will go to the lungs, and within the lungs, what do we know? These antigens, they will reach over here, and within the lung there is a presence of alveolar macrophage, right? Within the lungs there is a presence of alveolar macrophage. Now the alveolar macrophage, what they will do? They will start the process of phagocytosis. So here we will see the process of the phagocytosis. So if I just show these whole things over here, for example, so whatever the thing we'll see, for example, is the alveolar macrophage. So macrophage, and these are the antigens. And what for example, the antigens, the TB antigens over here. These are TB antigens. Now what do we know? In this way, during the phagocytosis process, they will what? These macrophages, they will secrete the lysosomal enzymes. So where we will get the lysosomes? We will get the additions or the fusions of the lysosomes along with these structures, and we will get the secretions of the different types of the destructive enzymes from the lysosomes. But as we know, the TB antigens, they are too resistant. They are too much resistant. So as a result, we will see this macrophage, they will be not able to degrade it. They will be not able to degrade, or they will be not able to destroy all the antigens. So we will see, as the TB antigens, they are resistant. So in these cases, we will see some antigens that will be not, for example, these antigens that cannot be degraded or that cannot be destructed. So whenever these antigens are not able to get destructed by the action of macrophage, whatever thing we'll see, as you know, within the chromosome number 6, there is special genes that is always producing the what? There is a special kind of the gene that is always producing the MHC class 2 molecules, right? MHC class 2 molecule. Now, whenever the MHC class 2 molecule that will be produced over here, and whenever the MHC2, that means also the major histocompatibility, or we can say the HLA2. So, I am just representing the terms in terms of the MHC class 2. So, MHC class 2 that will come over here, and then this MHC class 2 that will get bound along with the what this antigens. So, for example, I am just showing you the MHC class 2 molecules in this way, for example. Okay. Now, these MHC class 2 molecules, they will take the antigens and they will represent the antigens on the surface of macrophage. So, what type of thing we'll see? For example, these are the antigens. These are the antigens. And these antigens that will be represented by the MHC class 2 molecule that is produced by the chromosome number 6 on the macrophage. So, for example, we saw over here. So, this MHC class 2 molecule that will represent or that will express the tubercular antigen on the surface of the macrophage. That's why the macrophage, that is also known as what? Antigen presenting cell. That is also known as what? Antigen presenting cell. Okay. Now after that, what type of thing we'll get over here? That's the main thing. Okay. So up to now, I hope it's clear for you that at first the TB antigens they just came over here, and the macrophage, the alveolar macrophage, they did try their best to destroy all the tubercular antigens. But as we know that tubercular antigens are too much resistance. So, okay, so they cannot be destructed completely. So some of the antigens that will be left over here, and those antigens that will get bound along with the MHC class 2 molecule that will be produced by the chromosome number 6 that is present within the macrophage. And these antigens ultimately that will be represented or expressed on the surface of macrophage. Up to now, okay, this is clear, right? Now, what are the things we will get after that? Now, okay, let's talk in details about this one. See, whenever, try to understand, let me show over here, this MHC class 2 molecule, this MHC class 2 molecules on the macrophage that is representing the tubercular antigens, these whole parts, antigens, okay, and this is the MHC class 2 molecules, right? These are the MHC class 2 molecules. Okay, through the lymphatic system that will go to the lymph nodes, through the lymphatic regions, through the lymphatic ducts, we can write it over here, through the lymphatic ducts, okay, through the lymphatic ducts, it will go to the where? It will go to the lymph nodes. So ultimately we can write it over here, through the lymphatic systems, this whole thing that will be sent to the where? Within the lymphatic systems. And whenever it will come to the lymphatic system, whatever thing we'll see, as we know, within the lymph nodes there is the presence of what? T cells, right? We know there is the presence of the T cells. Now this whole thing, that means if I represent the macrophage over here, this is the macrophage, and if I represent the T cells, yes, so let's represent with this color only. Okay. Now what did you see over here? We just saw that within the macrophage there is the presence of tubercular antigens. The presence of the tubercular antigens, and along with that we also notice that there is also attachment of MHC class 2 molecules, that MHC class 2 molecules, for example. Now within the T cells there are special types of already genetically programmed. We will see that within the T cells there is the presence of a receptor for this type of the antigens, for tubercular antigens. Now this receptor that is known as, the receptors will get over here, that is known as, okay, let's show the receptor with this color, T cell receptor, that is known as T cell or receptors. Now we will see, along with these T cell receptors, along with the T cell or receptors, these antigens will get attached, and there is also another kind of the receptors, just to make sure that whether this antigen is attached with the MHC class 2 molecule or not. Just to check these things, we'll have different kinds of the receptors on the T cells just to check the MHC class 2 molecule. Now this receptor that is here for the receptors will get for MHC class 2, for example. We'll get the receptors over here for MHC to MHC class 2 molecules, and this is known as CD4. This is known as CD4 receptors, that is for MHC class 2 molecules. Now whenever these things will be attached together with these two things, after that we will get the different kinds of the co-stimulations, different kinds of the co-stimulator that will stimulate the what? T cells, right? That will stimulate the T cells. Now see what type of the co-stimulations we will get from the macrophage. We will get on type of the co-stimulator that is known as B7. So from here we will get another type of the co-stimulations that will reach over here, that is known as B7. And for the B7 we will have a different kinds of the receptor that is present over within the T cell, that is known as CD28. So we will get over here the CD28 for the B7. The B7 will get CD28. And another kinds of the stimulator molecule, or you can say the co-stimulator that will reach where within the T cell, that is known as interleukin 12. Interleukin 12. Now whenever we will see the stimulation square, at first the antigen is getting attached along with the T cell receptor, that is known as T cell or receptor, TCR. So T cell receptors. After that we are getting the attachment of the MHC class 2 molecule along with the CD4 receptors. After that we just saw over here there is also the additionals, or you can say the co-stimulators or the co-stimulations by the B7. And for the B7 that is produced by the macrophage, for just to take this thing, there is also presence of the receptors within the T cell, that is known as CD28. After that I just did the mentions of this thing, that there is also productions of the interleukin 12 that will go within the T cell, and that will also stimulate the T cells. And as we know, after the stimulations we will get within the T cells, there is the presence of another kind of the molecules just below this thing, that is known as CD3. So whenever this proper stimulation that will be raised to the T cells, this CD3 that will be also stimulated. So if I write it up, okay, let's CD3 that will be stimulated, and whenever the CD3 that will be stimulated, that will send the signals within the nucleus. That will send the signals to the nucleus. So from here we will get the signals to the nucleus. What are the things we will see within the nucleus? Let's talk about that thing. Now, if we see in terms of the from the CD3, whatever the thing we'll get is that T cells. Okay, that is the T cell. Now within the T cells, the signal that will reach up to the nucleus by the what? By the CD3. So you got over here the CD3. Okay, this is the CD3. And for example, it's the macrophage. We got over whatever I already explained you guys over here. Okay, is the macrophage over here. And whenever the stimulations will get over here, whenever the stimulations that will reach up to the nucleus, this on top of the gene that will be activated within the nucleus. So this gene that will be activated, whenever the first gene that will be activated, it will produce one kind of the protein that is, one kind of the chemical mediator that is known as interleukin 2. First it will produce what? The interleukin 2. After that, another kind of the gene that will be activated. Whenever this gene that will be activated, that will produce another kind of the protein that is known as interleukin 2 receptors. This is interleukin 2. Okay, let's draw it outside. We get the interleukin 2 or receptors. Now, whenever the interleukin 2 receptors were getting over here, the produced interleukin 2 by the first gene that will come across the interleukin 2 receptors, and that will get fused along with these structures. Now, whenever the interleukin 2 that will get fused along with the interleukin 2 receptors, the ultimate signal that will reach will be in the nucleus. And whenever the signal that will reach up to the nucleus, another kind of the genes that will be activated within the nucleus, rather the many genes will be activated. So these genes is having the capacity to produce interleukin 3. These genes also having the capacity to produce interleukin 4. And we also see the productions of the T1 necrosis factor. We also get the gamma interferon. Now we have to know on this thing, on thing over here, that whenever, how can we define the what type of the T cells it will be? It will be the Th1 or it will be Th2. How can we define this thing? For that thing, I'll just draw this part over here. First, we got over here this is the Th0, that is known as a naive cell. T naive cell. Within the T naive cells, within the nucleus, we'll get the different kinds of the genes. Okay. Now the T naive cells, that will make sure that whether it will be Th1 or whether it will be Th2, T helper cell 1 or T helper cell 2. What it will be, that will be determined by the naive cells. Now within the naive cell, within the nucleus there are different kinds of the genes, and the different kinds of the genes are responsible for the producing of the different kinds of the molecules. For example, this first gene, that whenever it will be activated, it will produce interleukin 2. After that, it will produce what? The interleukin 2 receptors. After that, it is producing the interleukin 3. It is producing interleukin 4. It is producing the tumor necrosis factor. It is producing the gamma interferon, for example. Gamma interferon. Now, if we see, okay, let's draw it over here, guys. Draw it over here. Now, if we see within the T cells, the T cells is getting activated by the presence of interleukin 2 that is produced by the macrophage. Whenever we get the stimulation of the T naive cells, for example, whenever we are getting the stimulation of the T naive cells by the interleukin 12, at that time, whatever the thing we'll get over here, let's talk about that thing first. So, in these cases, we'll get the gene, whatever the genes we'll see over here. The first gene that will be activated, that will produce the interleukin 2. Then the second gene that will be activated, that will produce, no problem, interleukin 2 receptor for the interleukin 2. After that, the genes that are responsible for productions of the interleukin 3 and interleukin 4, these genes will be inhibited. We'll get the dysfunction of this gene. We'll get the dysfunction of these genes whenever, try to understand what I'm saying, guys. Whenever the interleukin 12 that will activate, the T naive cells, at that time we'll see the genes are responsible for production of interleukin 3 and 4 that will be inhibited. So there will be dysfunctional. Rather, we'll see the expression of gene that is responsible for productions of the tumor necrosis factor and gamma interferon. What did I say, you guys? Now see, whenever we are getting the stimulation of interleukin 12 produced by the macrophage on T cells, at that time we will see this interleukin 12 that can switch off the gene that is producing the interleukin 3 and interleukin 4. Rather, this interleukin 12 that will help the T cells to express the gene to produce the interferon, gamma interferon and tumor necrosis factor. Okay, that's why whenever we get the inhibition of the genes for the production of interleukin 3 and interleukin 4, in this case, this type of the T cells is known as Th1. So this type of the cell that is known as Th1 cell. And what about this one? For example, in the same way, if we get okay, these T naive cells or the Th0 that is getting activated by interleukin 4 that is produced by the same cell, for example over here the macrophage. In these cases we will see, we'll get, try to understand, in these cases we'll also get the different kinds of the genes will be activated and some genes will be inhibited. So what are the things we'll get over here? See, the first gene that will be also able to produce the interleukin 2. There are also similarities in between the Th1 and Th2. Okay, and whatever the dissimilarities, I'll talk about all these things. Okay, then these genes will be expressed, these genes will be activated, they will express the molecules for the interleukin 2, that means interleukin 2 receptor. Then this gene will be activated, it will produce the interleukin 3, it will produce the next gene, interleukin 4, and the last two genes over here, that is responsible for the productions of the what? Tumor necrosis factor, and the lastmost gene that is responsible for the productions of gamma interferon. These two genes will be inhibited by the action of the interleukin 4 produced by the macrophage over here. Now, whenever we will get the activation of the gene that is able to produce the interleukin 3 and interleukin 4, and alone will get the that are responsible for the production of T1 necrosis factor and gamma interferon. These type of the cells are known as Th2 cell. That means T helper cell type 2. We can write it over here, Th2. Okay, and whenever we, as we know, the interleukin 3 and interleukin 4 that will be produced, these two things that can activate the B cell, and the B cell that will be able to, whenever we get the activations of that interleukin 3 and interleukin 4, and interleukin 3 and interleukin 4 that will activate the B cells, then the B cells that will be converted into the plasma cells, and these plasma cells they will produce the antibodies. And if we see overproductions of the interleukin 4, that will produce more amount of the IgE. You can remember this one, okay? Now, okay, let's come back to over here. As I said at first, the interleukin 2 that will be produced, and the interleukin 2 receptors, then we will get the productions of what? As in these cases, we will get the Th1, because in these cases I already explained you, there is activation of interleukin 12. See, there is activation of interleukin 12, the same thing. So it is supposed to be the Th1, the T helper cell type 1. Now in these cases, what? These two genes, that means the genes responsible for the productions of the interleukin 3 and interleukin 4, that will be inhibited. Rather, we will get the productions of what? T1 necrosis factor and gamma interferon. One more thing you have to remember, whenever the interleukin 2 receptor that will be produced and the interleukin 2 that will get bound along with the interleukin 2 receptors, the final signal, whenever the final signal that will reach where? Within the nucleus. After that, this signal that can proliferate, that is responsible for the proliferation of Th1. That is responsible for the proliferation of Th1. So here we will get the proliferation of Th1. Okay. Now we'll go back in details. What are the things we will get in the other things? Okay, in the other manifestations. Okay. Now, as we got, from here we will get the production of the T1 necrosis factor and the gamma interferon. What's their functions? They will look for the what? They will look for the TB antigens within the circulations. So ultimately, whenever we get the proliferations of the Th1 cells within the lymph nodes, so from the lymph nodes, these whole cells, you can say the colony of the cells, that will be taken by the what? That will be taken by the general circulation. So if I show it over here, guys, okay, now let me show these things over here. Now these whole things, we'll see, if we see clearly, guys. Okay. Now see these whole things, that means the whole thing, that means we will get the abundance of the Th1, within the circulation. At first it will go through the lymph nodes, and from the lymph nodes, from here it will go to the lymph nodes, and from the lymph node it will reach to the general circulation. Okay, it will go to the general circulation. Okay, let's continue. No problem. Now, whenever it will reach to the general circulations, what are the things we'll see? Now, for example, after 10 years or after 5 years, okay, there is the re-entry of the new tubercular antigens. For example, whatever the tubercular antigens that was, that came for the first time, for the initial exposures of the tubercular antigens, whatever the antigens was over here, we just took the medications, or by the immune systems, we were able to destroy all the antigens. Right now we are getting the new exposures of the tubercular antigens. So if we get the, okay, let's erase this part from here, guys. Whatever thing we'll get in this case. Now if we get the new exposure of tubercular antigen, whatever the thing we'll get. For example, these are the cells. Okay. So whenever we get the new exposures of the tubercular antigens, for example, these are the new tubercular antigens. Okay. Now whenever we get the tubercular antigens that will come over here, definitely the T helper 1 cell that will be activated, and these cells, let's show the different color this one. Otherwise it will be the same color, because I already drive the Th1 cell in the blue color. So let's make it red. So this is the newly exposure or new exposures of the tubercular antigens over here. Now whenever it will be definitely this stage 1 they are already programmed, they are already pre-synthesized for the tubercular antigens. Now we are getting the new exposures of the tubercular antigens. Definitely this stage 1 from the circulation they will rush to the cells, and whenever it will reach over here, try to understand whatever thing we'll see. These cells will go over here, they will get bound along with these structures. In the same way, we will get the all types of the stimulators, whatever I already explained to you guys. By that, we will get the productions of the interleukin 2, and then the interleukin 2 receptors. Then from here, we will get the ultimate signal, then we will get the clonal expansions, and from here, we will get the productions of what? The gamma interferon, and we will get the what? We will get the, okay, let's write it over here. Okay, it's clear. Now, along with that thing, okay, try to understand from here that whenever we are getting the gamma interferon and the tumor necrosis factor, what they will do. Let's talk about the first tumor necrosis factor and the other things, what they will do. Okay. Now, if we just draw the endothelial cellular lining, endothelial cellular linings over here. Now, whenever we are getting the productions of the endothelial cells, okay, what they will do, see, this gamma interferon, what they will do, let's write it over here, the gamma interferon that change the shape of endothelial cell. Now what they will do, they will make the endothelial cells, on the endothelial cells, they will produce the addition molecule for monocyte. Try to understand what did I say, you guys. The tumor necrosis factor. The tumor necrosis factor is having the capacity. Okay, let's draw it over here. Okay. Whenever the tumor necrosis factor that is acting over here, this tumor necrosis factor that will make the endothelial cells to express the addition molecule, for monocytes. So whenever these endothelial cells, they will express the addition molecule for the monocytes. So the monocytes, they will come over here and they will get fused. So whenever the monocytes, for example, the monocytes are coming over here and that will get fused. The monocyte will come over here, the monocyte is coming over here, that will get fused. In the same way, you get the fusions of the monocytes. Now another thing, the tumor necrosis factor that will do, that can stimulate the endothelial cell. Okay. It is supposed to be the tumor necrosis factor. So the tumor necrosis factor that can change the shape of the endothelial cells, because within the endothelial cells there is the receptor for the tumor necrosis factor. We will get the receptor on endothelial cells. Receptor on endothelial cell. So when you are reading the receptor on the endothelial cells for the tumor necrosis factor, they will change the shape of the endothelial cells, and they will make sure that the endothelial cells is making the addition molecules for the monocytes. So whenever the monocyte that will pass through the through the circulation, through the microcirculations over here, they will get fused along with the endothelial cells. After that, what other thing we get? This in the tumor necrosis factor that is also responsible for cause the vasodilation. Why? See, this tumor necrosis factor that will stimulate the endothelial cells to produce the prostaglandin. Okay guys, let's continue. So what I was saying you that the tumor necrosis factor is having the receptors on the endothelial cells. The tumor necrosis factor is having the receptors on the endothelial cells, and the tumor necrosis factor that will stimulate the endothelial cells to produce the prostaglandin I2, or you can say the prostacyclin. As a result, we will get the vasodilations over here. So more amount of the blood that will reach in the sites. Okay. Now after that, what type of thing we'll get? See, as we know that there is the productions of the gamma interferon along with the tumor necrosis factor. So see, let's write over here. This gamma, whenever this gamma interferon are the chemical attractors, or they can attract this gamma interferon, is having the capacity to attract the fused monocyte along with the endothelial cells. So whenever we see the concentration of the gamma interferon that will be increased, definitely we'll see the monocyte that will come out from the endothelial cells or from the circulations. So we'll get the end, the monocytes after coming outside the circulation, they are known as what? Macrophage. This macrophage, they will come outside. This macrophage, they will come outside by the chemoattracting property or by the attraction of gamma interferon. So whenever the gamma interferon is attracting the monocytes, so the fused monocyte along with the endothelial cells, by the attraction of gamma interferon, they will come out from that region and they will be within the site of infection. Now, whenever we'll see the concentration of the gamma interferon will be so much, we'll see this gamma interferon that can also change the shape of monocytes. Whatever the monocytes we will get over here, see, in these cases we will get the epithelial cells, epithelial cell shape, macrophages we will get over here. So this, as a result, we will get this type of the figure. We will get this type of the figure. Okay, and this nucleus, flattened, we get this like this. So at first, by the action of the gamma interferon, the fused monocyte along with the endothelial cells, they came out in the site of the infections. After that, whenever we will see the concentration of the gamma interferon will be so much that is also responsible for changing the shape of the monocytes, or you can say macrophage, outside the circulation. So we will see the shape that will be like the epithelial cell shape. Okay, so we can see it over here, epithelioid. Okay, write it over here, epithelioid cell. Now, along with that, we will see within these epithelioid cells, okay, whatever the thing we get, these epithelioid cells, right now these epithelioid cells, this is having the capacity to attract the lymphocyte. This is having the capacity to attract the lymphocytes. So from the circulations, we will get the lymphocyte that will come over here. For example, we are getting the lymphocytes. We are getting the lymphocyte. Further, we will see these epithelioid cells, by doing like this whole structures we are getting over here, this is known as, we are getting a large cells, this is known as our giant cell. We will get it, we can write it over here, this is known as giant cell. Further, we can get over here from these portions that, okay, here we will get for the productions of the more amount of the gamma interferon, we will get the fusion of these cells. And whenever we get the fusion of these cells, this is known as giant cells. Further, we will see these cells, they can also produce the different kinds of the growth factors. They can produce the different kinds of the growth factors. Whatever the growth factors they can produce, if we say in terms of the growth factor, in terms of the platelet derived growth factors, or they can also produce the fibroblast. So whenever they will be able to produce the fibroblast and the platelet derived growth factor like these things, we will get these whole things within the center. We will get the some caseous, or the whitest part within the center. We will get this region, sometimes this is known as caseous. So we will get, in terms of the tubercular infections, we can get the caseous necrosis, that is also known as caseous necrosis in these cases. Okay, hope it's clear for you guys. Now I'll repeat this thing once again. Try to understand this thing very clearly. At first, try to understand, as I said, that the tumor necrosis factor is having the receptors on the endothelial cells. So whenever the tumor necrosis factor that will activate the endothelial cells or stimulate the endothelial cells that will produce the prostaglandin I2, or it is also known as prostacyclin. As a result, we will get the vasodilation. The first thing, okay. Second thing, we will get over here, this T1 necrosis factor is also responsible for the productions of the additive molecule for monocytes on the endothelial cells. So the T1 necrosis factor is doing first function. The T1 necrosis factor is having the receptor on the endothelial cell. As a result, the T1 necrosis factor that can stimulate the endothelial cells for the production of prostaglandin I2, or you can say the prostacyclin. As a result, we will get the vasodilatations. The next thing we will get over here, the T1 necrosis factor that can also change the shape of the endothelial cells. How? The T1 necrosis factor that will make what? The receptors, or you can say the adhesion molecule on the receptors, or on the what? On the side of the endothelial cells for the monocytes. Now, whenever we get the more amount of the gamma interference, where the site of the infection, so more amount of the, we'll get the outcoming of what? These monocytes. Whenever the monocytes, they will come out of the circulations, they will be known as what? Macrophage. So whenever we get the macrophage over here, then we'll see this, the more, if we see the concentration of the gamma interferon is so much, that is also responsible for changing the shape of macrophages into the epithelioid cells. And whenever we get the accumulations of the epithelioid cell along with the lymphocytes in the whole structures, this is known as giant cell. In this case, we will get the fusion in between the two epithelioid cells, and that will make a large complex that is known as epithelioid cells. Now, in which type of the disease we can get this type of the thing, we'll talk about in the next videos, and we'll also talk about the different kinds of the test by which we can also represent the type of hypersensitivity reactions. Okay. So this was all about in terms of the first part. In the next part, we will talk about what type of the disease. For example, we can get the multiple sclerosis. We can get the dermatitis. Okay, this how they are developing this type of the disease for the presence of type 4 hypersensitivity, and how the cells are involved over there. We'll talk about all these things in the next part. Thanks for watching. See you in the next video soon.